These are the found into the three combination is called the nucleotide. Coming to the next one, nucleic acids. Nucleic acids are two. One is the one is a DNA, another thing is the RNA. Okay, RNA and DNA both are present in this one. Next one, another thing is called, next point is called one genetic code is believed to call up one of the 20 different amino acids. How many amino acids to present in this one? 20 different types of amino acid. How many are there? 20. 20 different types of amino acid during protein synthesis, and therefore there must be 20 types of genetic codes. Genetic codes. One nucleotide cannot act as one genetic code. See, listen careful. One genetic code, one nucleotide cannot act, one genetic code cannot act one genetic code. That because they are only, because they are only one amino acid cannot act as the one nucleotide. Okay, one nucleotide cannot act the genetic code because they are only four nucleotides. What are the four nucleotides? Only four nucleotides, whereas the amino acid for which they have act as codes are 20. The nucleotide to act as genetic codes therefore must act in multiple unit. Multiple unit. Nucleotides act as in unit. How many nuclei act as in unit? Only two. The possible dinucleotide combines. That is called the twice. They will be, you can calculation by the 4 into 4 or 16. 4 into 4 are 16. Therefore, there are few to account of these 20 amino acids. If the nucleotides are then assumed to be arranged units of their the possible trinucleotide combination tripled, that is called the trinucleotide, that is called the triplet, that is called triplet. Triplets, triplets. That is name is called triplets. Will be, that is you can calculate like this, 4 into 4 into 4 is equal to, or 64. 64 are shown in the genetic code number. This is the genetic code. Okay. This is the genetic code. Approximately the 64, this one. Approximately the 64 possible triplets are too many for 20 amino acid. These are possible if 20 amino acids is there. These possible triplets are too many for 20 amino acid. Overlapping triplets have been suggested to cut down the number of possible trinucleotide to somewhere about 20. Okay. Several experiential evidence, including that of uh, Korhana, the scientist name Korhana, have, however, differentially established that there is no overlapping of triplets and that they act separately. The problem of the great number or triplets has been solved by postulating the view that in the case of some amino acid, more than one triplet, say two or even as many as six, may act as one code. See, listen careful. The amino acid, uh, the some of the amino acids, more than one triplet, say two or even as many as six, may act. One amino acid to act one, many, two or six, act as a one code. Such genetic codes are termed as degenerate codes. What is the name of the code? Is called degenerate code. The code has been described as degenerate because the same amino acid is coded by more than one base triplet. Okay, these genetic code main systematic nucleotide sequence of mRNA, the degenerating particle amino acid sequence DNA, the code words amino acids is called, this is called codons. Okay, coming to the next one. 1 second, this is not moving. See, 1 second. Okay. After that one, first you can observe these codons. Okay. Next, coming to this one. See, these are the codons. The degenerate is there, no? The codon, you can observe the table. 4 into 4 into are 64 as shown in this table. How many are there? See, you can observe here UAC, U meaning uracil, C meaning cytosinin, A meaning adenine, uracil, cytosinin, adenine, guanine. These are the nucleotides, okay? Purines and pyrimidines. These are the purines and pyrimidines. Cytosine, stop. These are these. Okay. Coming to the degenerate, the code has been described as degenerate because the same amino acid coded by more than one base triplet. Degeneracy has been established in the genetic code by natural selection as a mechanism for boosting bolstering stability against the effect of random mutations. If the code were not degenerate, what is the meaning of if the code were not degenerate? That is only 20 triplets out of 64 specified the 20 amino acid. Most of the one-step mutations would lead to the triplets that would not specify the amino acid. The results would lead to unfinished polypeptide chains expressed in the organism as defective mutations. Okay. The next problem is breaking of the code. What is the breaking of the code? That is breaking. These are the You can observe the table showing nucleotide combination, 16 nucleotides, AA, AC, GA, GC. This is a 16 types nucleotide tides twice. 16 twice is the 64. Total triplet nucleotide chart. You can observe this one. What is the 64? 64 is possible codons, nucleotides, adenine, guanine, cytosine, uracil. Triplet base 4 threes are 64, like this. Okay. The next problem is the breaking of the code. How to breaking of the code? What is that? Is that meaning to find out which triplet stands for which amino acid. The first major breakthrough in the study of the triplet dictionary came in 1961 when at the 5th International Congress of Biochemistry in Moscow, a young biochemist from the National Institute of Health, Marcel Nirenberg, scientist, reported the experiment on protein synthesis in vitro condition. His result amazed the scientific world. He had made the breakthrough which lead directly to deciphering of the codon. Nirenberg associates. Then Nirenberg and his associates to explain that one. What is the Nirenberg and associates to explain? Nirenberg associate Harich took an artificial RNA in their experiment. These two scientists how to experimented to that one. First I'll explain. After that one, I'll explain to the what is universal, what is that one. Here, Nirenberg and his associates took an artificial RNA in their experiment, which had which had only one type base that is called uracil. See here A adenine, adenine, uracil, adenine. Uracil you can all observe only RNA, but DNA cytosine is present. In the place of uracil, you can observe to the cytosine. These are the basics, purines and pyrimidine base structures. Okay. This uracil, the RNA was thus all poly-U. The two workers succeeded in synthesizing a protein containing only one type of amino acid. What is the amino acid? Phenylalanine is the one type of amino acid. The first amino acid scientists to experimentally identify. What is the amino acid? Phenylalanine amino acid in the word of Nirenberg and Mithai. What is the name? Nirenberg and Mithai. The addition of poly-U resulted in the incorporation of phenylalanine alone into a protein resemble polyphenylalanine. That's where you can write down poly-U appearance to function as a synthesis template or messenger RNA. Synthetic messenger or messenger RNA. In this system, one or more uracil acid residues appear in this code. That is called phenyl UUUU. That name is called phenylalanine is called UUUU. And become the first word. The first word is codon. The first word is called codon. These codon to be discovered in the genetic dictionary. Since then a number of artificial RNA polymerase. Number of artificial RNA polymerase have been synthesized containing every possible combination of the four bases. they have they have been tested for their ability to synthesize protein in vitro condition. The another scientist name is called H.G. Corona. Corona. This is scientist name, very, 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 very important. This scientist, he is associate at the University of Wisconsin. The university name Wisconsin. Has been very useful in this directions. In these directions, by the end of 1960, the total assignment of the 64 triplet codons was completed. How to codons to scientists to divisions, all things I'll explain. Okay. It was the complementary but independent work Marshall, Nirenberg, Corona, three scientists, and Robert Holly. These are scientists. First one, Marcel, W. Marcel. Second scientist name, Nirenberg. Third one, Hargovind Corona. Third scientist, H.G. Corona. They call the name is called H.G. Corona, meaning Hargovind Corona. And Robert W. Holly, which lead to the breaking of the genetic code. For their brilliant work, they were jointly awarded the Nobel Prize. They were jointly awarded to the Nobel Prize for Medicine and Physiology in 1968. The amino acids and their corresponding triplets are given the table. See this in this table, you can observe the all or the present in this table. See this one. It is evidence that 61 of the 64 triplet specify amino acid. The three remaining. What are the three? See, you can observe the red color is there. Here 64 are there. In this 64, the evident that 61 of the 64 triplet amino acid. Specify amino acid. The three remaining triplets. See what is the remaining. You can observe the in the color chain UAA, UAG, UGA. These three do not specify amino acid. How many codons are there? Totally 64 codons are present, but only 61 codons specify the amino acid. Remaining three do not specify the amino acid. What are they? These are called UAA, UAG, UGA. These three codons are called stop codons, or another name is called nonsense codons. What is the name? That is called the nonsense or stop codons. Okay. How many total to found in that one? Only 61 codons code with 20 type of amino acid. Remaining 3 is not coded by the amino acid. These three name UAA, UAG, UGA. Okay. This is called nonsense codons or nonsense codons, but act as signals for terminating polypeptide chain. But this one also useful, but it is not useful. These three codons also very, very useful codons. These codons to stop the terminating polypeptide chain. In protein synthesis time, the chain to forming the chain polypeptide chain, when the processing is completed, in that time, these nonsense codons to signal the polypeptide chain to stop. That is called the terminating codons. That is called, what is that one is called? terminating codons, terminating polypeptide chain. Okay, chain growth is initiated by the codons GUG, AUG, but initiation codons. What are the initiation codons? GUG. First one is the GUG is the first codon. AUG. See, is there no AUG? GUG. Second one GUG. Where GUG? G. See, this one GUG. This one. This is the first. What is the names here inside? Here names are there, no? Yes or no? These are, okay, these codon, this amino acid code. That is the names. Here red color codon, red color picture is there, no? In that red color picture, that is called the nonsense codons, stop codons, not to the any amino acid to not, but here these are the useful function is there. What is the useful function? They have stopped the polypeptide chain, but that's why this also used. But chain growth initiation codons also too. AUG, GUG. See, AUG, methionine start codon. This is the valine start codon. Then two are, okay? They occur at the beginning of mRNA. Sorry, where you can observe in this one. They occur beginning in the mRNA of following a stop. That one is the stop. The codon specifics methionine both at the beginning of the protein and also internally the codon GUG normally specific valine. See here in the chart in the board you can observe this is the codon GUG normally specify which amino acid, valine amino acid. However, this GUG occurs at the beginning of mRNA. I'll explain mRNA time. Occur at the beginning of mRNA or following a stop. It code for the methionine. It code for the methionine amino acid. A study of triple would show that the first and second base in the triplet are more important than the third in the third in distinguishing amino acid. This is more evident in the Crick and Obel scientists to explain. Which scientists to explain? Crick and Obel. Now you can observe to the another slide, protein synthesis. Now how to character see 64. These are things I'll explain now. Universal. A few AUA codes for methionine in mitochondria. AUA ILE in cytoplasm. UGA stop codon in cytoplasm. UGA triplet in methionine. In bacteria, GUG, UUG, AUG, AUU, CUG, initiation codons. These are all initiation codons. Stop codons. What are the stop codons? Initiation codons. Starting codon AUG. Stop codon 3. UAA, UAG, UGA, UAG, UGA, UAG. These three are the stop codons. These three are stop codons. Expecting codons in 21st century. That is AA. This is one selenocysteine. That is called the selenocysteine UGA. The genetic code is called degenerate. Now, if I'll explain, it is unambiguous codon. That is unambiguous codon. See, this is the codon total. This is the first letters, third letters. These are the first and your U is the first, third. U C, A, G, like this. Okay. Hmm. What is the codon is overlapping? Overlapping without punctuations, meaning comma, full stop, not like that. You can write down like this. Obeling phenomena. Now I will explain. No, a study triplets would show the first and second base in triplet are more important than the third is distinguished amino acid. This is more evident for the crick and world of the obel upside. They reduced strategy between the third base of codon and complementary base action. In mitochondria have different codes are there. This is the obel the phenomena or obel scientist formula. Now I will start to the protein synthesis. What is the protein synthesis? What is the importance of protein synthesis? Protein biosynthesis involve a number of steps which are outlined. First, before protein synthesis, what is the activation of the amino acid? Activation of the amino acid. It was for a long believed that only activated amino acid could bring about protein synthesis. The scientist Hoagland in 1955 was first one identified activated from of amino acid working on protein synthesis in rat liver. This scientist working on which animal? The rat animal liver. Hoagland scientist found that amino acid and ATP reacted to produced and activated from of amino acid. Only after 2 years, Hoagland, Jeminick, and Stephenson discovered that a small tRNA present in the soluble fraction of cell was also needed in the process of amino acids. Okay. Here, it has been found that to form amino acid transfer RNA complex, each of the 20 amino acids must be activated before these are attached to their respective transfer RNAs. The activation of amino acid takes place by its reaction with ATP. When amino acid is formed by phosphorylated released, the reaction is catalyzed by specific amino enzyme, that is the amino acyl tRNA synthetase. This means that at least 20 enzymes must function at this stage. Here, magnesium also involved in the reaction. The adenine is bound with the enzyme and remains so unit it is transferred transferred to the particular RNAs. This is the importance of the uh, amino activation of amino acid. Now I will to explain to the translation. Translation, transcription, these three are most important for the protein synthesis. Before translation, we will discuss about to the 1 second. One second, I'll explain that one. What is the transcription? What is the translation? These are things we will discuss now. Okay, before protein synthesis time, what is the transcript? What is the transcription? How to transcription process are present in this one total processing. These are things we will discuss now. 1 second. Okay, now translation is defined as a protein synthesis. Translation is defined as a protein synthesis occurs in ribosome mRNA. RNA also three types of RNAs are there. One is the mRNA. Second one is the rRNA and tRNA. Messenger RNA, transcription RNA, tRNA, transfer RNA, rRNA, ribosomal RNA. rRNA is called the ribosomal RNA. These are RNA's type. Now, the transfer of amino acid here occurs in ribosome mRNA to protein. You know, ribosome to mRNA translated in 5' to 3' direction, highly regulated, very fast. Yeah, these things before I'll explain that one. The tRNA react with cytidine tryptophase, that is called the CTP. Cytidine tryptophase is called the CTP, and adenosine tryptophase ATP, adenosine triphosphate, cytidine tryptophan or cytidine triphosphate, CTP ATP. CTP cytosine, ATP is adenosine triphosphate. Mm. Before amino acid is before amino acid is transferred, transferred to it, transferred to it. This transfer, these two or transfer reaction is also catalyzed. These also catalyzed by the same synthesis. Catalyzed by the same synthesis enzyme which catalyze the reaction between amino acid and ATP. Adenosine triphosphate is the involvement, and adenosine second one, adenosine monophosphate, that is called AMP. AMP meaning adenosine monophosphate, and amino acyl synthetase are released in this reaction. The adenylene derivative is transferred to a hydroxyl group of the three-terminal adenine nucleotide. This is a tRNA. There are at least 20 different amino acid tRNA synthesis for specific binding between the two types of amino acids and 20 types of tRNAs. For certain amino acid, there may be more than one tRNA species, as many as the tRNA ribosome 80S tRNA. Specific species have been identified. The species name is called E. coli bacteria. The species name is called the E. coli. The species name is called the E. coli bacteria. E. coli bacteria, and the next one for certain, in that one certain bacteria, these all things to explain to the 30 specific as certain amino acid may be more than tRNA species. 80 type is that tRNA have been identified the E. coli. The number of synthetase are, however, less than one the number of tRNA. That is the same activating enzyme might be shared by several tRNAs. Might be shared by the several tRNAs. This is the translation. In the translation, see, this is a tRNA. Transfer RNA stripe. In the cytoplasm, this is the ribosome, 80S ribosome. This part, in this one attached the tRNA. This red color part is the messenger RNA. See, these are tRNAs, okay? This is a ribosomal subunits. You can observe to the ribosomal these are all the ribosomal subunits. And another thing is these are all, say, these are all the ribosomal things. These are all the ribosomal things. This one is the mRNA. 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 One second. This one is the mRNA method, and these are all the tRNAs. Template mRNA, tRNA that you know, that is called the transfer RNA. tRNA is called the transfer RNA linked to the amino acid. Ribosomes, you know, tRNA ribosomes is the 80S type of ribosomes are there. And next one is the many accessory protein and some energy GTP hydrates. Okay. Next one is the another one is the initiation. Some energy GTP hydrates are there. See, function of the types RNA, mRNA serve as a template code. tRNA serve as an adapter molecule. rRNA hold molecule in the correct position. Protein proportion also catalyze. See, in this picture you can observe this is the mRNA. This part is the mRNA. This is the tRNA. This one, these all the clover leaf model is the tRNA. mRNA, tRNA. See, these are the ribosomal RNA. These three are most important. Hold the protein rRNA and in the correct position. Protein portion also catalyzed reaction. The translation in most biological process in eukaryotes. Five stages are there. Pre-initiation. First one is the translation is the most complicated biological process now. In eukaryotes, 70 type ribosomal protein, 20 more protein to activate as a 12 more auxiliary enzymes involvement is there. 100 proteins for processing. 40 tRNA and rRNA, transfer RNA and ribosomal RNA. Other specific proteins are involvement is there in this process. 300 molecules also to found in this one. Before the protein synthesis, here 5 stages are there. One is the pre-initiation stage. Initiation stage. Second one is the initiation. Third one is the elongation. Fourth one is the termination. And fifth one is called post-termination modification. Post-terminal translation modifications. Post-transitional modification. First one, pre-initiation. What is the pre-initiation? See, pre-initiation changing of AAA, amino acid. Say, this is a tRNA, transfer RNA model. This is the amino transfer RNA clove leaf model. AUG is the amino, AEs is the attaching amino acid for 3' to 5'. These are the three anticodons. These charging amino acid linking amino acid to correct tRNA, catalyzed amino acyl tRNA synthesis, that is AAtRNA. These are the total finally coupling of 20 different syntheses. Two steps are involvement. That is called, that is the activation of amino acid. And second one is the transfer of amino acid to tRNA. These are the charging to tRNAs. Okay. Now we will discuss to the tRNA how to initiation process to present in this one. It has been observed that a particular site of 30th subunit of ribosomal gets attached to the bringing end of the thread-like messengers. Here, two thread-like messengers are formed in this one. According to many scientists or many workers, scientists, the transfer RNA through to attach the itself of here the ribosomal two type. One is the 30S and second one is the 50S type. 30S and 50S both are to the involvement is present in this one. Okay, of this ribosome. The site of attachment must be highly specific, chemically. This is grouping formed. This grouping formed is the ribosomal surface by ribosomal RNA and the structural proteins of the ribosome together. It has been discovered in the prokaryotic cells. Prokaryotic, the binding site of the mRNA are the first 3 to 8 bases here, which are complementary to the 3' and bases of the 16S type of RNA, smaller subunit, that is 16S smaller subunit. This complementation between the base mRNA and rRNA, RNA, essential for correct starting of the protein synthesis. This is also helped by the fact that the secondary structure to tRNA, that is the loops of the allergen or mRNA proper side. This one, you can observe this picture. The it is in this one prokaryote, the protein synthesis. You can observe the total formation of initiation complex. How to initiation activate to the tRNA and mRNA. In prokaryotes, the protein synthesis begins with the special kind of initiator that is called tRNA. Listen careful. I'll explain to them the prokaryotic cell only. Okay. In the different are there. That prokaryotic, the protein synthesis begins with begins with the protein begins with the special kind of a initiator that is called tRNA, which has the formulated methionine F-met tRNA. It is well known that the amino group and carboxylic group COOH group amino and carboxylic group of two amino acid combined together in peptide linkage. The aim of F-met or tRNA, that is a methionine tRNA, which has the NH2 group blocked by the formal group, is to keep only COOH group for free for linkage to the NH2 group of the next amino acid. This ensure correct sequence of amino acid linking together. It is interesting to note that AUG in any other part of the mRNA can code for only the normal methionine and not the formulated one because of certain factors. GUG, which is a code. GUG, which is a code for valine. Before slide, I'll explain no the code, which code is which to is present. Valine is there. This valine code for F-met is present as initiator codons. Three initiator codon factors termed IF1, IF2, IF3 and GTP. IF1, IF2 and GTP required the initiation codons. Next one, the in eukaryotic. Now I will complete the prokaryotic cell. Now eukaryotic cell. The eukaryotic cells in the cytoplasm, the initiation is brought about by a special met tRNA. You know met tRNA. Met tRNA, met tRNA is called methionine tRNA. Met tRNA is called the methionine tRNA. Methionine tRNA, this methionine tRNA, which is not formally formulated. The factors of initiation in eukaryotes are much more complex in nature. They are IEF1, IEF2, IEF3, F4, 5, 6, like this. Beta, F2. These are the phosphorylated by specific kinase will not be activated unless it is phosphorylated by another enzyme, a cyclic AMP depending protein kinase. This is the initiation period. Second one, elongation. See, formulation of the GTP, EFI, and I'll explain these all things now. Okay. The second one, see, HCR, PKR, these are things I will explain. No, next one, coming to the second one, elongation process. Next second one, EF1 control, and the next one is the elongation process. 1 second, I will explain to the elongation. This one. Elongation of polypeptide chains. The elongation of the polypeptide chain begins with the arrival of new amino acyl. See in the picture, this is the ribosome. Okay, mRNA, this part is mRNA. This is the this is a tRNA. How to attach? One added to a at a time, one at a time. The elongation process. See this one. The elongation process. The elongation of the polypeptide chain begins with the arrival of a new amino acyl tRNA on the amino acyl or acceptor site of the 50S. See, this is the 50S subunit of the ribosome. 50S subunit of the ribosome. It is not clear whether the first tRNA, the FMET tRNA, comes to the A site or the peptidyl or donor site of the 50S subunit. Even it comes to the A site, first it to the transferred the P site before the next tRNA attached to the A site. The next part in the elongation process is the formation of peptide bond. The amino acid of the first transfer RNA is now linked by its free amino group to the free carboxylic group peptidyl tRNA at the P site. See, this is the P site. This one is the P site and A site. This one is the A site. P site, in the presence of enzyme, peptidyl transferase enzyme. The tRNA, see you can observe the tRNA. The tRNA of the P site is now deacylated so that the dipeptide is linked to the tRNA of the A site. This is the A site. Now you can observe here. This is the P site. This one is the above one is the A site. P and A site. Now the ribosome is supposed to move ahead 5 prime to 3 prime directions. 5 prime to 3 prime directions of the mRNA. The supposed of the mRNA. 5 prime to 3 prime of the, see here mRNA is there. mRNA, this one, 5 prime to 3. At this time, the peptidyl tRNA, see, which one tRNA? The peptidyl, see in the picture, the peptidyl tRNA of the A site is transferred to the P site under the influence of GTP. Which influence are there in this process? GTP influence, vanillin triphosphate influence in GTP and transfer factor G. Which factor 2 is there here? G, that is the translocase. That one name is called translocase. Meanwhile, the GTP and factor G8 is release of the previous tRNA which move into the cytoplasm. See, the cytoplasm. And next one, previous cytoplasm to pick up another specific amino acid molecule. See, this one totally process. This one is a totally process. Entire A site amino acid tRNA. See, this one, this part, how to one by one to attach to move, like this. Okay, when the ribosome has moved far enough, another ribosome can attach. Okay, did you understood this point? When the ribosome has moved far enough, another ribosome can attach itself to the starting end of the same RNA and so on. These ribosome will make identical copies of the same protein since they are reading the same genetic message. That is illustration by varying length of polypeptide chain, which have been synthesized by different ribosomes at different stage during their movement along the messenger RNA chain. Amino acids are added at the rate of 2 per second. 2 per second. This is the repeated energy or the to form. That is called the transcription. Translation, growing peptide chain move from the A site to P site like this. A site, this one, P site. Deaccelerated tRNA moves out from P site to E site. E site, this site. Exit site is the E site. This is the year the total energy to gives from the GTP. This modal is for the GTP. And next one, process of peptide synthesis occurs until a termination codon is reached. This is the, what is the step? This one, elongation step. Next one, coming to the next one, termination step. Very, okay, this process is going on. But where you can stop the codons to stop? This termination peptide chain at the end of the polypeptide chain can chain synthesis and enzyme bring about hydrolytic splitting of the last peptide tRNA, last peptide tRNA ester linkage, release the polypeptide chain, and the ribosome is disassociated from the mRNA. This is brought about UAA. You can observe this one. UAA, UAG, UGA. These nonsense codons, these three is called nonsense codons, and the termination factor, R1 factor, R2 factor. The ribosome is also disassociated with the ribosome also disassociated into subunits by the factor IF3 factor. IF3 factor. The requirement of proteins. The requirement of protein synthesis are shown to this one. The whole process of the protein synthesis is symmetrically represented. That one is a total symmetrically. This is the termination process. Regulation protein synthesis. This is the termination process. See, the stop codons should be turned by the processing. See, these are the activation of the termination codon. One second, this one. Stop codons appear A site. Here, no tRNA not available. Releasing factor, releasing factor RF1 recognize site presence in A site. The RF1, RF3, GTP. These three hydrolysis of bond between peptide and tRNA occupying P site. That is a water molecule is added. Protein tRNA 60S and 40S ribosomes, that is a GDP is released. See, this is a bioenergetic. This activation of AAA to high energy bond, entry of amino acyl tRNA A site, 1 GTP also. Translocation of peptidyl tRNA from A site to P site, releasing here 1 GTP, 5 energy bonds. In prokaryote, how much of time to take here? In prokaryotic, 18 seconds to take the time. In eukaryotic, only for the 6 seconds time. Only for the how much of time to take a 6 type. This is a polysome in eukaryotic even. In eukaryotics, the same molecule of mRNA can be simultaneous transformed several times. Each emerging peptide is synthesized on a separate ribosome. Many ribosomes on the same starting of mRNA are called polysomes. See, ribosome 5 prime to 3 prime end. Ribosome first. Second, polypeptide chain to elongation. Cheaper proteins to form. And final, protein synthesis. And next one, terminate of the this one. Okay, got it? This one, P bond. Here, non-transpirating mRNA, P bond site, non-translating mRNA can form to the ribonucleoproteins particles, that is a mRNP, accumulate in the cytoplasm organelles as the P bodies, that is called the P bodies. P body site for translation represent and mRNA decay. 35 distinct proteins are found in P bond alike mRNA decapping enzyme, RNA helicases, exonuclease, etc. Few mRNA related. Environmental factor, regulation in translation, iron exchange, synthesis, virulase also until these all things are for level protein synthesis like this. Primary structure, this one. Secondary structure, tertiary, quaternary. Tertiary, this is the alpha helix, beta globin polypeptide, beta globin polypeptide tensor. And chevrons and protein folding, these are the main important source, the inhibitor. Before inhibitor, the protein synthesis, the protein regulation of protein synthesis, main is the regulation protein synthesis is extremely important for an organism since it enable to it adopt itself changing environment and phenomena growth. It was known as for long that certain microorganism were capable of adopting their enzyme influence. This is phenomena as enzyme adaptation or induction. Absolutely. There was two types, insuitable enzyme, consecutive enzyme. These all are the types of protein synthesis. Now, these are the, one second. This is the the ribosomal inhibitor translation. Reversible inhibitors are there. Tetracycline, that is the antibiotics. It is also most important protein synthesis. Which one is the inhibitors are there? Irreversible inhibitor protein synthesis, tetracycline. Binding to tetracycline is the antibiotic. That is just like to the antibiotic. This one, tetracyclic binds three inhibited 30S type of ribosome attached of amino acyl tRNA to A site. Next, chloromorphonisol inhibit. Why these are you can many time diseases are there. In that time you can use for the antibiotics. No, in that time the antibiotics to act as the in the bacteria or viral or anyone to stop the protein content, that is up. That is the inhibitors. For this is the inhibit peptidyl transferase. Next, erythromycin. You know, in this infection, throat infection, fever, viral infection, you can use for the these antibiotics. These antibiotics is the tetracycline, erythromycin, cylindromycin, prevent in the translocation. Very important. Inhibitors also most important. Irreversible inhibitor. Some are irreversible inhibitors also. Streptomycin aminoglycosides. Streptomycin also one type of this one. What is the that is called the antibiotic. This is amino acyl. This one is amino acyl Aminoglycosides bind to the 30S ribosomal subunit. Low concentration, misreading of protein, useless bacteria proteins. Pharma conclusion inhibitor. This is a synthesis. Protein synthesis is inhibitors. Next one, puromycin. Another one is a mammals, inhibitors in mammals. Puromycin, structural analog, triosinal tRNA. Triosinal tRNA. Next one, cycloheximide. Cycloheximide inhibitor peptidyl transfers. Diphtheriotoxin inactivation of EF2 by attachment of ADP to EF2. This is the ricin also inactivated 20S type of rRNA. Thank you. Today class we will discuss about total genetic code and protein synthesis. Okay. Now we will start another class that is a different topic. Wait some time. Nitrogen metabolism and protein synthesis. Nitrogen metabolism, you have completed to the total biological nitrogen fixation, nitrogen biological nitrogen, asymbiotic nitrogen, genetic nitrogen denitrogenase, biochemistry, genetic code, central dogma, transcription, concept of protein synthesis, translation, regulation of protein synthesis. These are things are completed. Uptake of sulfur, reduction of sulfur, you can observe. Now another class, we will discuss about to the, one second, uptake of the next one, we will discuss about uptake of sulfur. When one second, I'll show the, hello, hello, చెప్పాలి. మంచి Hmm. Now we will discuss about to the uptake of the sulfur. Uptake of the sulfur, how to uptake of the sulfur. Sulfur is also, uh, 1 second, I'll share my PPT. Uh, Uptake of the sulfur. Sulfur is also one of the essential elements and used in the synthesis of amino acid like cytosine, methionine, sulfur, cytosine and methionine, methionine, cytosine and methionine. Sulfur plays a structural and regular roles as disulfide bridges in protein. Sulfate is relatively abundant in native soils, and its deficiency is very rare. Sulfur is observed as sulfate by plant root from the soil. The gaseous form of sulfur, SO2, sulfur dioxide, enters the leaves. Sulfate is abundant in areas which air pollution, and the source of sulfur is significant in these areas. Sulfur is transported as sulfate. The sulfate must cross three membrane systems. The plasma membrane of root cell at the soil plant interface. The plasma membrane of the internal cells. The plasma membrane of internal cells involved in a transport and the cytochloroplast membrane. Within the chloroplast, it is reduced to sulfide. It is also transported across the tonoplast into the vacuole. Sulfate may be reduced to sulfide and is converted into thiol group. This may be incorporated into cytosine or converted in thioester, methyl, sulfonin compounds, and sulfoxide is there present in this. The uptake of sulfate transfer, the sulfur is taken as in sulfate by active coke transport mechanism into a plant root along with protons. The stoichiometry is shown to be 1 is to 3. 1 is to 3 method is the sulfate and protons. The proton gradient is maintained across the membrane by the proton pumping ATPase. Proton pumping ATPase. See, across the material, increase in pH of the external medium inhibited, sulfate uptake. The sulfate transported are shown to be localized in plasma membrane. Plants are known to contain multiple transporters with different affinities for sulfate as relieved by experiment at molecular level. Sulfate uptake activity is increased by sulfur starrations and repressed by supply of reduced sulfur. Because protein synthesis is necessary for induction of transport activity, it is assumed that the experience of transporter is regulated. The transport of sulfur into vacuole differs from that plasma membrane. in that is mediated by unipotor. The vascular sap has high acidic pH due to activity of tonoplast of the ATP age. Tonoplast of the ATP age. How to transportate of this one? The transporter of sulfur into the vacuole differs from that plasma membrane in that is mediated by unipotor. The vascular sap has high acidity pH due to the activity of tonoplast proton ATPase. This steep electrical gradient is used by the unipotor to transport sulfate from cytoplasm to vacuole. Sulfate transport into plastid is considered to be through several transports like triose-phosphate translocator, proton sulfate co-transport. The sulfate uptake is inhibited by sulfide, selenite, molybdate, chromate, and chromate as the chromate with sulfate binding with the transfer. High and low. See, high efficiency and low. High and low efficiency sulfur, high and low efficiency, the sulfur, the sulfur sulfate transport proteins are encoded by a gene family. A high affinity isoform is expressed in roots and the transporter content of the protein is increased under starvation. A low affinity isoform is expressed predominantly in leaves and less responsive external supply of sulfur. Low affinity types mediate transport within the sulfur. The sulfur reduction is the activation, reduction of sulfide and incorporation of sulfide into cytosine. First one is the reduction. The reduction of the sulfur. The reduction of the sulfate. See, primary step. The reduction of the sulfate to sulfide is an energy consuming process requiring the energy 732 kilocalories energy required for this obtained from ATP, and the reductant is derived from photosynthesis in green tissue. In non-green tissues, these may be obtained from respiration or oxidative pentose pathway. The enzyme for cytosine biosynthesis are located in chloroplast and root plastid. The activation assimilation of sulfate ion begins with activation. Sulfate is activated in presence of ATP with the help of the enzyme ATP resulting in the formation of 5-adenylyl sulfate. The forward reaction is favored by the conservation of APS and PP1 to other compound. Two enzymes, APS kinase, APS reductase, are also known as APS sulfotransferase metabolism. APS and PP1 is hydrolyzed to inorganic phosphate by inorganic pyrophosphate. The enzyme ATP sulfurase is purified from spinach leaf and its properties were studied. It exists two forms. Isoform, one is the chloroplast occurring 85 to 90%. The second 90% of the total enzyme and the other in the cytoplasm. Two types of ATP sulfurase has been identified from various organisms. One heteromeric enzyme regulated by endotrionic GTP activity prokaryotes. Second one, homoric enzyme without regulatory GTP activity from the eukaryotic. How to reduction of the sulfate? The reduction of APS takes place in plastid. Takes place in the plastid. APS reductase transfer two electrons from reduced glutathione to produce sulfate. So sulfite is reduced. Sulfite is reduced to sulfide in the presence of sulfite reductase. The sulfite reductase transfer to the 6 electrons from reduction pyridoxine. Pyridoxine in the sulfide. See, you can observe this picture. This picture you can observe. The result sulfide then combined with the acetylserine OAS acetylserine to form cysteines and acetate. The O-acetylserine is formed from serine and acetyl-CoA in the presence of serine acetyl transferase enzyme. These uh, transport amino pathway, the reduction of APS exclusively occur in the plastid, whereas sulfonation of APS takes place in cytosol. APS is the first phosphorylate in the presence of APS kinase to form 3' phosphate 5' phosphosulfate PAPS. PAPS is then reduced to free sulfite and ultimately sulfide in presence of the enzyme thyroxidine depending PAPS reductase and pyridoxine depend sulfite reductase respectively. The ratio of reduction sulfate nitrate in plants maintained at 1:20. APS reductase activity declining under nitrogen starvation. OAS is the regulator molecule. It upregulated sulfate permease and APA synthesis coordinating sulfate reduction and cysteine synthesis. This is the total alternative pathway of the sulfate. This is the sulfate high efficiency sulfate transport also present in the plants. Next to carbon, hydrogen, oxygen, nitrogen is the most essential element. It is a consequently chlorophyll, proteins, nucleic acids. Nitrogen is a colorless and odorless gas. Most of the nitrogen is obtained from soil as nitrate to ammonia. The reduction of dinitrogen known as dinitrogen fixation. There are several nitrogen fixing organism, asymbiotic, symbiotic, and blue-green algae. Proteins are macromolecule with one or more polypeptide chains. The mRNA synthesized from DNA and based on the message to protein synthesis. Next one, sulfur is also most essential. Sulfur is also most essential element and it is used in the synthesis of amino acid. The sulfur is taken as sulfate. Increase in pH of the external medium inhibit sulfate uptake. The sulfate transport consists of both constitutive S nutrients depending regulate transport. A decrease intracellular concept sulfate, cysteine and glutathione is associated increasing in transport of activity. The gene and protein expressions studied have confirmed that regulation occurs predominantly at the level of mRNA. A breakdown the expression pattern of the gene sulfate transfer family reflect a complex pattern regulation. The first pattern, expression of the gene, that is the group 3 transport with tissues, organisms specific but not to regulation by S nutrients. The second one, cell specific expressions of some group 1, group 2, group 4 transporter under adequate S nutrients with is upregulated by inadequate S nutrient. Cell tissues specific S deficiency related depresent of the some group 1 and 2 transported. This is the more sulfur how to use for the translation and transport the whole plant of the useful of the sulfur. Now these all things I'll stop my sharing. Now these all things are today class we will discuss about to the all things meaning all meaning what is nitrogen biological nitrogen fixation, symbiotic nitrogen fixation, asymbiotic nitrogen fixation, and physical nitrogen fixation. Physical meaning industry level how to nitrogen to form that one. And the biological in that one, biological nitrogen fixation. And the next one is the protein synthesis. Protein synthesis meaning the protein synthesis, what is the genetic code? How many genetic codons are there? How many amino acid is present? How many code with the genetic amino acid? Here 64 genetic codons there. 20 types of amino acids are present. These 64 codons to code with the 20 amino acid. Each and every codon minimum 3 to 4 or 5 types of amino acid code with that one. This in the protein synthesis central we will discuss to the central dogma also, central dogma, and the next one after central dogma proteins in the what is the translation, what is the transcription, what is the initiation codon, what is the termination codon, what is the these are things we all discuss today class. And other last one sulfate also, sulfur useful how to transport the plants, what is the importance of sulfur, these are things we will discuss today class. Okay, thank you.